Hello everyone. Welcome to another rapid review lecture by the UIMS. My name is Arian Taniwall and I am a fifth-year medical student at the second faculty of medicine at Charles University. Today we'll be talking about endocrine physiology. These are the sources that I have mainly used for this lecture. First of all, the Guyton, and second of all, Costanzo physiology book. Before I begin, I would like to say that this lecture should be used by you as a form of supplement to the other sources of information that you use and the other books that you have for your particular subjects. All right, let us begin. So this is the basic outline of our today's lecture. We're going to first start with the different forms of chemical signaling, and then we will start with the endocrine system. We will talk about the chemical characteristics of hormones, as well as how they actually work inside our bodies. And then we will talk about the hypothalamic pituitary axis, as well as the hormones secreted by the anterior and posterior pituitary glands. All right, so before I actually start talking about the endocrine system, we should talk about the different forms of chemical signaling, basically different routes for the signal molecules inside the body to travel from the source to the target receptor. And the main difference between these different types of chemical signaling are basically the distances that the signal molecules have to travel to reach their targets. So the first type is the autocrine signaling, where the signal or the signaling molecule targets the cell which has secreted that particular signaling molecule, which means that the cell targets itself. An example of this signaling can be found, for example, during embryological development, where this form of signaling ensures that a large number of multipotent cells develop into the same type. And another example of autocrine signaling would be seen with viral infections, where a cell which has been infected with a virus starts secreting molecules which will induce apoptosis of that same cell. The second type of cellular signaling is the gap junction signaling. And this basically involves targeting a cell, which is a neighboring cell and which is connected to the cell which produces the signaling molecule via a gap junction. So we can see on this figure right here that the signaling cell produces a particular messenger molecule which diffuses to the target cell via these gap junctions. An example of this form of signaling would be seen in cardiomyocytes, where this form of signaling ensures synchronized contraction of the myocardium, as gap junctions will ensure that the depolarization or the wave of depolarization spreads from one cell to another in a synchronized manner. Another form of cellular signaling is the paracrine signaling, where a cell targets a nearby cell. This means that the signaling molecule diffuses through the extracellular fluid and reaches a target cell which is in a relatively close range relatively to that signal producing cell. A subtype of this paracrine signaling is the synaptic signaling, where a neurotransmitter diffuses from the presynaptic end of the nerve fiber to the postsynaptic membrane. And a paracrine signaling usually produces quick responses which last only a short amount of time. Finally, endocrine signaling, which is the one we're actually going to talk about, involves production of a messenger molecule, in this case a hormone, which travels in the bloodstream to reach a distant cell. So this form of signaling can reach cells all over the body. However, it produces slower responses, which are actually longer lasting. All right, so now we can actually start talking about the endocrine system. And this is a messenger system composed of feedback loops of the hormones released by internal glands and hormone-producing cells of an organism directly into the bloodstream, which regulate the physiology of distant target organs. On this figure right here, we have illustrated the various endocrine glands in the body, from hypothalamus to the thyroid gland, adrenal gland, and so on. The main function of the endocrine system is to maintain homeostasis, a stable, constant internal environment of the body. And it works along with the nervous system to achieve that. So it has many functions, and one of, or a few of them, would be regulation of the growth and development, regulation of reproduction, blood pressure, electrolyte balance, as well as behavior. And because I said that the endocrine system cooperates closely with the nervous system, we should compare these two systems next to each other and see what the differences are to better understand the endocrine system. So the communication method, as we know in the nervous system, is a nerve signal and action potential, which is transmitted from one nerve fiber to another via the synaptic cleft, via synapses. While in the endocrine system, there are hormones which are secreted into the blood and transported to cells throughout the body. Now, the target of stimulation in the nervous system are other neurons, muscle cells, and glands, but in the endocrine system, it can actually be any cell in the body with a receptor for that particular hormone. There are also differences in the response time, as well as range of effect and duration of response. So usually the response time in the nervous system is rapid, while in the endocrine system, it takes longer time. As far as the range of effect, with the nervous system, it's usually localized and only affects specific sites in the body, but with the endocrine system, it can have a much more widespread effect. And the duration of response, I already mentioned this, with the synaptic signaling, it's usually short-term, while with the endocrine system, the response to the action of the hormone can last days to weeks and may continue even months after. So let us talk about the general principles of endocrine regulation. How is the endocrine system regulated? So in order to maintain the homeostasis of the body, secretion of hormones must be regulated and turned on and off as needed. And this regulation is maintained by two main mechanisms. It's either the neural mechanism or feedback mechanism. The neural regulation involves direct stimulation of the endocrine cells by neurons. And we can see this on this diagram where the neuron sends an action potential, which is then transmitted to the synapse and to the endocrine cells. And at the synapse, there's a stimulatory neurotransmitter, which acts on the endocrine cell and stimulates it to secrete a particular hormone into the bloodstream. And a classic example of this form of regulation of the endocrine system would be in the case of a short-term stress response, where we have preganglionic sympathetic nerve fibers, which synapse on the cells of the adrenal medulla and stimulate it to release catecholamines like epinephrine and norepinephrine into the bloodstream during some stressful situation. But a more common and widespread mechanism of regulation of the endocrine system is the feedback mechanism. And this basically involves some element of the physiologic response to a hormone, which feeds back either directly or indirectly on the endocrine gland, which actually secreted the hormone, and this will change its secretion rate. This means that it's either the hormone or the response that it causes, which then acts back on the glands, which have actually caused the secretion of that particular hormone in the first place. And it can be either positive or negative. The primary type is negative, while positive feedback is relatively rare. With the negative feedback, it basically involves some feature of the hormone action or the hormone itself, like we can see here, which then negatively acts on the upstream glands which cause the secretion of that particular hormone. So we can see in this example, we have this hormone testosterone, which can negatively feed back on the anterior pituitary or on the hypothalamus. And it can decrease production of the hormones by these two glands, which will then lead to decreased stimulation of the endocrine gland, in this case testes, and will lead to decreased production of that hormone, testosterone. And there are different types of feedback loops. There are long feedback loops, short feedback loops, as well as the ultra short feedback loop, where the hormone inhibits its further secretion. This would mean that a hormone produced by the anterior pituitary gland acts on the hypothalamus. The hormone produced by the anterior pituitary gland acts on the pituitary gland itself and will decrease the secretion of that particular hormone. Basically, the net result of this is that if the hormone levels are adequate or high, the negative feedback loop will prevent further secretion of the hormone, while if the hormone levels are low, secretion of the hormone is stimulated. And on the other hand, we have the positive feedback, and this means that some feature of the hormone action or the hormone itself increases secretion of that same hormone. And this is a self-augmenting process. It means that in this case, for example, we have the hormone estradiol, which positively feeds back on the anterior pituitary gland and causes it to secrete more hormones which stimulate the endocrine gland, in this case ovary, to produce estradiol. And this positive feedback loop is self-augmenting, leading to some explosive event, we could say. Two examples of this form of positive feedback. The most common ones are during the midpoint of the menstrual cycle, estrogen has a positive feedback effect on the secretion of FSH and LH by the anterior pituitary gland. And this positive feedback effect leads to ovulation. And another example is the positive feedback of oxytocin. I will talk about oxytocin later on, but basically what happens here is that oxytocin will cause dilation and contraction of the uterus, which actually stimulates increased secretion of oxytocin by the posterior pituitary gland. And this positive feedback loop leads to increasing contractions of the walls of the uterus, and eventually leads to birth. Okay, so now that we talked about the main ways how endocrine system is regulated, let's talk about the chemical characteristics of hormones. So there are basically three main types of hormones. There are peptides, steroids, and derivatives of amino acids. So on this figure over here, we can see that there are actually four types listed, but the peptide and protein hormones are usually listed as one type. So the peptide hormones, these are basically synthesized by combining short or long chains of amino acids together. Steroid hormones are derived from the lipid cholesterol, and amine hormones are basically derivatives of amino acids with modification of their groups. For example, the amine norepinephrine. And certainly there are going to be differences between the various features of these hormones. And these are listed on this table right here. So as I already said, there's going to be a difference in the synthesis. So peptide hormones or protein hormones are going to be synthesized in a very similar way to the way that other proteins are synthesized in the body. So there's going to be a specific sequence, a genetic sequence, which is transcribed into an mRNA, and the mRNA is translated and packaged in the Golgi organ into secretory vesicles, and then it is secreted when the particular endocrine cell is stimulated. While in the case of the steroid hormones, they are synthesized from cholesterol, and these amines are synthesized from the amino acid tyrosine. The storage also differs between these different types of hormones. So the peptides can be stored in vesicles. I mean, yes, the peptides are stored in vesicles, while the steroids are usually not stored, but they are released immediately when needed. So this is called constitutive secretion. Amino acid derivatives are stored before release, but the storage mechanisms vary. Most of the peptide hormones are polar and water-soluble. They bind receptors on cell membranes and then transduce the signal via the use of second messenger systems. And they often cause fast changes in the protein activity through gene expression changes. Examples of these peptide hormones are the hormones of the pituitary gland, such as prolactin, ACTH, but also other hormones like insulin, glucagon, and parathyroid hormone. As far as steroid hormones, these bind to intracellular receptors and they will directly change expression. And I will talk about this mechanism of action further in the next slides. As far as the effects of steroid hormones, they will alter the gene expression. And even though they have a slower onset, they have usually a longer duration of action than the peptide hormones. Examples of steroid hormones are cortisol, aldosterone, so these are adrenal cortex steroids, as well as the sex steroids, estrogen, progesterone, testosterone. And amino acid derivatives, some of them are polar, others have to be bound to proteins in the serum. And as far as their action on receptors, adrenaline, epinephrine acts on the membrane receptors, while the thyroid hormones, they act directly on the nuclear receptors. As far as effects, adrenaline functions like peptides, while thyroid hormones function in a similar manner to steroid hormones. And the examples of amino acid derivatives are adrenaline, thyroxine, triiodothyronine, so the thyroid hormone. Now, as far as the mechanism of hormone action, let's first talk about the peptide hormones, and this also applies to catecholamines, to epinephrine and norepinephrine. So the first step is the hormone binding to the membrane receptor on the target cell. Then there's a formation of a hormone receptor complex, which is coupled to a G protein. This is a GTP binding protein, and this G protein is coupled to a particular effector protein. Most commonly, these are enzymes adenylyl cyclase or phospholipase C. These are the two main mechanisms of the peptide hormone mechanism of action, either the adenylyl cyclase mechanism or phospholipase C. But of course, there are other mechanisms as well. When the effector proteins are activated, a second messenger, cyclic adenosine monophosphate C AMP, is actually produced. Okay. And this will then lead to activation of downstream protein kinases, phosphorylation of proteins by that activated protein kinase, and it will finally orchestrate the final physiologic action. It is worth to note that muscarinic receptors are also coupled to G proteins, and they also utilize these G protein mediated mechanisms, such as the adenylyl cyclase or phospholipase C. So this is a figure explaining the adenylyl cyclase mechanism. Again, the hormone binds to the receptor. We have formation of the hormone receptor complex. And the receptor is coupled to a G protein, which when activated by hormone will bind GTP, and the GTP will activate adenylyl cyclase, which will start producing cAMP from ATP. And the cyclic AMP activates protein kinases, which leads to phosphorylation of these downstream proteins and orchestrates this final physiologic action. And this is the second mechanism, the phospholipase C mechanism, where hormone binds to the receptor. We again have the formation of the hormone receptor complex and activation of the G protein. But in this case, the GTP, which binds the G protein, activates phospholipase C. This is membrane-bound enzyme, which will actually catalyze the conversion of PIP2 into inositol triphosphate and diacylglycerol. So the diacylglycerol will directly increase activity of protein kinases, and the inositol triphosphate will increase the release of calcium from endoplasmic reticulum to the cytoplasm, and this increased intracellular concentration of calcium will activate protein kinases. And then protein kinases phosphorylate downstream proteins, which then cause the final physiologic action of that particular protein. Those were the peptide hormones and also catecholamines, as well as the neurotransmitters at muscarinic receptors. Now, as far as steroid hormones and thyroid hormones, they work differently. So they bind either to a cytosolic or a nuclear receptor. And then the receptor hormone complex basically becomes a transcription factor. So it is translocated into the nucleus. And this complex of the steroid hormone and the receptor uses its zinc fingers to attach to the DNA of the cell. The particular genetic sequence is called the steroid response element, and it will induce transcription of that particular gene. And then we have production of a particular protein, which is involved in causing the physiologic action of that particular steroid hormone or thyroid hormone. And as I already mentioned before, the contrast to peptide hormones is that they act relatively quickly on their target cells within minutes, but steroid hormones take longer, usually taking hours at least. Okay, so those were the basics about how the endocrine system works, how it's regulated, and what are the underlying signaling processes of the various hormones. But now let's talk about the different glands of the endocrine system. Let's talk about the hypothalamic-pituitary system. So these two glands, hypothalamus and pituitary gland, function in a coordinated, synchronized fashion to regulate many of the endocrine subsystems. The HP system regulates function of the thyroid gland, adrenal gland, reproductive glands, and it also controls the growth of the body, milk production, as well as osmoregulation, regulation of the osmolarity of the plasma. So the pituitary gland consists of the anterior lobe, called adenohypophysis, and the posterior lobe, neurohypophysis. And the hypothalamus is connected to the pituitary gland via the infundibulum, the stalk, which involves or facilitates the control of the pituitary gland by the hypothalamus via both neural and humoral mechanisms. Okay, so let's talk about the relationship between hypothalamus and posterior pituitary gland. The connection between them is purely neural. In fact, the posterior pituitary gland is a collection of axons whose cell bodies are located in the hypothalamus. We can see it on this diagram right here, that we have the cell bodies in the nuclei of the hypothalamus, which then extend their axons all the way to the posterior pituitary gland. It's also illustrated on this picture right here. So the hormones secreted by the posterior lobe, which are ACTH and oxytocin, are actually neuropeptides. They are peptides released from neurons, from those endings of those axons, which reach the posterior lobe of the pituitary gland. And the cell bodies of the ACTH hormone and the oxytocin secreting neurons are located in the supraoptic and paraventricular nuclei within the hypothalamus, respectively. Once the particular hormone is synthesized in the cell body of the hypothalamus, the hormones are then transported down these axons and stored in nerve terminals in the posterior pituitary gland. When there is a need for one of these hormones, the cell body of the neurons in the hypothalamic nuclei is stimulated, and this leads to release of the neurosecretory vesicles from the nerve terminals by exocytosis, and it leads to secretion of the hormone into the nearby capillaries so that the hormones can actually enter the systemic circulation. So here we can see the capillaries, which will then into which the hormones will empty. And on the other hand, anterior lobe and the hypothalamus have a different relationship. So the connection between them is both neural and endocrine. This means that there are certain hypothalamic releasing or inhibiting hormones. They are called liberins and statins, respectively. These are synthesized in the cell bodies of the hypothalamic neurons. These hormones then travel down the axons of these neurons to the median eminence of hypothalamus. And when these hypothalamic neurons are stimulated, hormones are secreted into the surrounding hypothalamic tissue at that median eminence, and they enter the nearby capillary plexus. These hormones then enter these capillaries, drain through the long hypophyseal portal vessels to the anterior lobe of the pituitary, and the hypothalamic hormones then act on the endocrine cells of the adenohypophysis, and they either stimulate or inhibit the release of those adenohypophysis hormones. So I will explain that again. So we have the cell bodies of neurons in the hypothalamus, in the various nuclei of hypothalamus. The hypothalamus secretes liberins or statins, those releasing or inhibiting hormones, into the median eminence right here. These hormones are then secreted from the nerve endings of these axons, and they enter the circulation. They enter these capillaries right here, and through the long portal vessels, they enter another plexus of capillaries in the anterior pituitary gland. And here they act on the endocrine cells of the anterior pituitary gland, and they either stimulate these cells or inhibit them. If they stimulate them, this will lead to secretion of hormones of the adenohypophysis, which can then enter the capillaries and enter the bloodstream. Okay, so let's talk about the hormones of the anterior pituitary gland. So the adenohypophysis secretes 6 peptide hormones, which are illustrated right here on this diagram: prolactin, TSH, LH, FSH, growth hormone, ACTH. I will talk about each one of these hormones later. So each hormone is actually secreted by a different cell type, with an exception of the FSH and LH, which are secreted by the same cell type. So here I have written which types of cells secrete these particular hormones, and the percentage corresponds to the percentage of the total endocrine cells in the adenohypophysis, which is occupied by that particular cell type. Okay, so one more note. On this diagram, there is the prolactin releasing hormone, but actually this is a hypothetical hormone. It hasn't been proven to exist, so that's why I have crossed it out. But other than that, this diagram is correct. So we can see that, for example, here we have lactotropes, which produce prolactin, which acts on the breasts, thyrotropes producing TSH, which acts on the thyroid gland, and so on and so forth. And here we also have the hypothalamic hormones, which either are inhibiting or releasing. Okay, let's start with thyroid-stimulating hormone. This hormone facilitates the major control of the synthesis and secretion of thyroid hormones via the hypothalamic pituitary axis. And the two main factors, or the two main processes which regulate TSH secretion, are the TRH, the thyrotropin releasing hormone from the hypothalamus, which stimulates secretion of TSH by the adenohypophysis, and thyroid hormones, which inhibit the secretion of TSH. And they do this by downregulating the receptors for TRH on the thyrotropes on the adenohypophysis. So basically, what happens is that, as I said, two ways TRH can act on the thyroid tropes to increase secretion of TSH, or the thyroid hormones themselves, they act on the pituitary gland and they downregulate receptors to the thyrotropin-releasing hormone. What are the actions of thyroid-stimulating hormone? So this is a peptide hormone which will bind to a membrane receptor and then will act through an adenylyl cyclase mechanism, which I already explained in the previous slides. And this will lead to increased synthesis and secretion of thyroid hormones by stimulating every step in the synthetic pathway of thyroid hormones. And it will also induce hypertrophy and hyperplasia of the thyroid follicular cells. And it will also increase the thyroidal blood flow. So this is actually the reason why in Graves' disease, in an autoimmune disease where we have autoantibodies against the TSH receptor, why this is the reason why it can lead to goiter formation, because we're stimulating the TSH receptor. So it's almost as if this TSH was acting on the thyroid gland to induce the hypertrophy and hyperplasia. Well, in this case, it's just an autoantibody. Okay, let's talk about the next hormone of the adenohypophysis, the ACTH or Adrenocorticotropic hormone. The way it works is that it stimulates the synthesis and secretion of adrenal cortex steroids. So it increases activity of cholesterol desmolase. This is a very important enzyme involved in the rate-limiting step of synthesis of steroid hormones, so the adrenal cortex. But it also increases transfer of cholesterol to mitochondria where the steroid hormone synthesis will occur. I think it's important to note that ACTH mainly regulates the synthesis of glucocorticoids and adrenal androgens, while the mineralocorticoid synthesis is mostly controlled via the renin-angiotensin-aldosterone system. Okay, now ACTH secretion. It is stimulated by corticotropin-releasing hormone, secreted by the hypothalamus, which will stimulate corticotropes to secrete ACTH into the bloodstream. And it is also important to note that ACTH has a pulsatile and diurnal secretory pattern, which will drive the parallel pattern of cortisol secretion, meaning there's a peak in the cortisol secretion in the early morning, and then there's a gradual decline to the lowest level at midnight. And this is actually caused by the parallel pattern of secretion of ACTH. There's also negative feedback exerted by cortisol on the hypothalamus as well as on the adenohypophysis, which means that when there is a chronic deficiency of cortisol, it will lead to stimulation of the hypothalamic pituitary axis, and this will lead to increased ACTH levels. And this is actually used in diagnosis of the various causes of hypofunctioning adrenal cortex to differentiate whether it is a peripheral cause or whether it is a central cause, whether it's the adrenal cortex or the pituitary gland which is affected. There will be difference there. Okay, but this will be covered more in the pathology lectures. Okay, let's talk about growth hormone, also called somatotropin. This is the most important hormone for the normal growth to the adult stature. It's secreted in a pulsatile pattern every day, with bursts of secretion occurring every 2 hours. Greatest secretory burst occurs about 1 hour, within 1 hour of falling asleep. And the secretory rate also differs depending on what stage in our life we are. So there is a gradual increase of the growth hormone secretion from birth into childhood. And then at puberty, we have the highest level of growth hormone, this enormous burst, which is actually induced by testosterone in males and estrogen in females. And it will cause increased frequency and magnitude of these repeating secretory pulses or secretory bursts of growth hormone during the day, and the main effect is that growth spurt during puberty. And then it will, growth hormone secretion will gradually decline until the lowest levels in senescence. Okay, so how is growth hormone secretion regulated? Basically, we have the, again, hypothalamic hormone, growth hormone releasing hormone, which acts on the somatotrophs of the adenohypophysis to induce greater transcription of the growth hormone gene and increase secretion of the growth hormone. And then, on the other hand, there's somatostatin, another hypothalamic hormone which inhibits growth hormone secretion by blocking the action of growth hormone releasing hormone on the somatotrophs. There are other factors which can stimulate or inhibit growth hormone secretion. For example, exercise or stress will increase the growth hormone secretion to help the body fight the particular stressful situation. Fasting, starvation, as well as decreased glucose concentration, all of these will increase the levels of growth hormone. While on the other hand, inhibitory factors, I already said senescence, and also hyperglycemia, among others. So what are the actions of growth hormone? Growth hormone has multiple metabolic actions of the various organs of the body, and it basically promotes growth of virtually every organ in our body. The actions of growth hormone are either mediated by the direct effect of the hormone on the target tissue, or it is mediated indirectly through production of somatomedins, insulin-like growth factors in the liver. So the growth hormone acts in the liver, and that's where these insulin-like growth factors are produced, which then act on other tissues. So this is an indirect action. So the three main actions of growth hormone are increasing insulin resistance, meaning there's going to be decreased glucose uptake and utilization by the muscles and adipose tissue, as well as increased lipolysis, which means that growth hormone will basically increase glycemia. It will increase the energy available for the growth metabolism. It will also increase protein synthesis and organ growth in virtually every organ. There's going to be increased uptake of amino acids, synthesis of proteins and DNA, and overall it's going to lead to an increased lean body mass and increased organ size. The increased lean body mass is obviously something that athletes strive for. So there's quite a big problem with the growth hormone doping in athletes, such as wrestlers, bodybuilders, martial artists. But the problem here is that it's going to lead to increased organ size, and it can lead to problems such as hypertension, it can lead to hypertrophic cardiomyopathy, and even heart failure, as well as other organ defects. And finally, it causes increased linear bone growth, meaning that it will basically, this is the main hormone which causes the achievement of the adult stature. It will increase the proliferation of chondrocytes at the epiphyseal plate, and it will increase the rate of growth in height, linear bone growth. Okay, another hormone of adenohypophysis is prolactin. This is a hormone which is responsible for milk production as well as development of the breasts during puberty. In a non-pregnant, non-lactating person, blood levels of prolactin are low. However, during pregnancy and lactation, these increase. The levels of prolactin increase. Now, the regulation of prolactin secretion, it is stimulated by TRH. As I said before, the prolactin releasing hormone is a hypothetical hormone. It hasn't been proven to exist yet. And the TRH, the thyrotropin releasing hormone, actually acts on the lactotrophs and increases the transcription of the prolactin gene to increase the secretion of prolactin from adenohypophysis. On the other hand, prolactin secretion is inhibited by prolactin inhibiting hormone. This is dopamine. And the reason why in non-pregnant and non-lactating persons there are low levels of prolactin is because these inhibitory effects of dopamine dominate and override the stimulatory effects of the TRH. Now, during breastfeeding, what happens is that the suckling stimulates the afferent fibers at the nipple, and this carries information to the hypothalamus, and it actually inhibits secretion of dopamine from the hypothalamus, and it will decrease the inhibition of prolactin secretion so that more prolactin can be produced and there is greater amount of milk produced during breastfeeding. So what are the main actions of prolactin? As I said, it causes milk production. It will stimulate the milk production by causing transcription of the genes responsible for enzymes involved in synthesis of the main components of the breast milk, such as lactose, casein, protein, as well as the main lipids. And it will also cause breast development. So prolactin will actually stimulate proliferation and branching of the mammary ducts during puberty. And third of all, it inhibits ovulation. So this is the reason why there is decreased fertility during breastfeeding. Prolactin inhibits ovulation by inhibiting the synthesis and release of gonadotropin releasing hormone, which means that this will basically lead to decreased production of the luteinizing hormone and follicle stimulating hormone, decreasing the fertility of the person. Okay, finally, last two hormones of adenohypophysis, FSH and LH, follicle stimulating hormone and luteinizing hormone. So these are also called gonadotropins, and their main functions are that they regulate the steroid hormone synthesis, sexual development, and reproductive function. So the follicle stimulating hormone, in males, it maintains optimal spermatogenesis and function of the Sertoli cells, while in females, it acts on the granulosa cells and induces the optimal follicular development as well as synthesis of the estrogens, the main estrogen estradiol. Luteinizing hormone stimulates the Leydig cells in the testes to synthesize testosterone, and in females it stimulates ovulation and development of corpus luteum, which will secrete progesterone to facilitate implantation of a fertilized egg. All of these processes will be more thoroughly explained in the lecture about the reproductive system. So as far as the secretion of the FSH and LH goes, it is stimulated by the gonadotropin releasing hormone, which is a hypothalamic hormone, and it varies over the lifespan. So here we can see how the secretion of FSH and LH varies over the life. So there's an increase through the puberty into the adult reproductive period, where there's actually a pulsatile, this oscillating secretory pattern, which is actually caused by pulsatile secretion of gonadotropin releasing hormone, which then drives this parallel secretory pattern. The pulsatile secretion of FSH and LH will drive the testes and ovaries to secrete their respective sex steroid hormones, and increasing circulating levels of these sex steroid hormones are then responsible for the appearance of the secondary sexual characteristics at puberty. Finally, in some sense, gonadotropin secretion actually increases further. However, because the gonadotropin releasing hormone secretion is so high, it will lead to downregulation of the receptors for this gonadotropin releasing hormone in the anterior pituitary gland. So actually the consequence of this is decreased production of FSH, decreased production of the sex steroid hormones in old age. Yes. I would like to also talk about how the FSH and LH secretion is regulated during the menstrual cycle. So during the first 14 days of the menstrual cycle, this is the follicular phase, midpoint is the ovulation, the last 14 days are the luteal phase. So during the follicular phase, there is a negative feedback of estradiol, the main estrogen, on the secretion of gonadotropin releasing hormone and FSH and LH secretion by the anterior pituitary gland, which we can see right here. There's a negative feedback. But at the midpoint, estradiol levels will rise sharply because there's a proliferation of the follicular cells in the ovary. And once a critical level of estradiol is reached, it will actually start having a positive feedback effect on the hypothalamus and anterior pituitary gland. So this is an example of that positive feedback which I talked about before. And this leads to a self-augmenting process, and it leads to a burst of hormone secretion, which will eventually lead to ovulation. And then in luteal phase, the major hormone secreted by ovaries will be actually progesterone and not estrogen, and it will have, again, negative feedback effects on the hypothalamus and anterior pituitary gland. And here we can see nicely how there, at the ovulation, there's a peak of the estrogen, and then peak of LH and FSH, which leads to ovulation, and then these hormones gradually decrease their levels, and progesterone rises because the corpus luteum produces the progesterone to facilitate implantation of the fertilized egg in the uterus. Okay, so those were the hormones of the anterior pituitary gland of adenohypophysis. Now let's talk about hormones of neurohypophysis. So the neurohypophysis is derived from neural tissue, and it secretes two peptide hormones, ADH, antidiuretic hormone, and oxytocin. Now these both act on their respective target tissue, the kidney and arterioles for ADH, and the breast and the uterus for oxytocin. Now, these hormones are synthesized in the cell bodies of the hypothalamic neurons, which are located in the nuclei of the hypothalamus, and then they are secreted from the nerve endings in the posterior pituitary gland. So even though both hormones are actually synthesized in both the supraoptic and paraventricular nuclei, the ADH is actually primarily associated with supraoptic nucleus, and oxytocin is primarily associated with paraventricular nucleus. So the secretion of these hormones is initiated when an action potential is transmitted from the cell body of the hypothalamus down to that axon in the neurohypophysis, which will stimulate release of these hormones from the nerve terminals into the capillary plexus. Okay, so let's talk about ADH, antidiuretic hormone, also called vasopressin. This is the main hormone responsible for regulating the osmolarity of the blood. So, what are the factors which will cause secretion of ADH? The main one is increased serum osmolarity. This will be sensed by the central osmoreceptors in the anterior hypothalamus, and then action potentials will be propagated down from the hypothalamus, down the axons to neurohypophysis to increase the secretion of the ADH, which will cause increased reabsorption of the water, as I will explain later, and will then decrease the serum osmolarity. Another process which can stimulate secretion of ADH is hypovolemia. So we have decrease in the extracellular fluid volume, and this then leads to decrease in the mean arterial pressure. This will be sensed by the various baroreceptors in the body, and the information is transmitted to the hypothalamus via vagus nerve, and the hypothalamus will then again direct increased secretion of ADH at those nerve terminals in neurohypophysis. More water will be reabsorbed, and the extracellular fluid volume will be restored. There are, of course, other stimulatory and inhibitory factors which can influence the secretion of ADH. An interesting one is ethanol. So ethanol actually has inhibiting effects on the secretion of ADH, which explains its diuretic effects on the body. And as far as actions of the ADH, antidiuretic hormone go, there are two main actions. One of them are the kidney, and the other on the vascular smooth muscles. So in the kidney, vasopressin causes increased water reabsorption by binding to the B2 receptor. It causes increased insertion of these water channels called aquaporins into the membranes of the tubules, luminal membranes of the tubules, and this will increase the reabsorption of water and will decrease the blood fluid osmolarity, as well as it will increase the extracellular fluid volume. And the second effect that vasopressin has is on the vascular smooth muscle. It causes contraction, and this will lead to constriction of arterioles, which leads to increased total peripheral resistance, and this then leads to increased mean arterial pressure. So actually vasopressin can be used in the cases of bleeding, for example, gastrointestinal bleeding, to promote constriction of these arterioles and decrease the loss of blood. Now, the second hormone of neurohypophysis is oxytocin. So this hormone is secreted by various stimulating factors. So what can cause secretion of oxytocin is suckling or sight, sound, or smell of the infant, as well as dilatation of the cervix. The suckling itself, as I already said, it stimulates the efferent nerve fibers of the nipple, and then this information is transmitted to the hypothalamic nuclei, which will cause release of the oxytocin from those nerve terminals at neurohypophysis. So what does oxytocin actually cause? First of all, it causes milk ejection from the mammary glands. It causes contractions of these small myoepithelial cells which line the mammary ducts, and this will then force the milk into the larger ducts and eventually will lead to flow of the milk through the nipple and out of the breast. Another function it has is the uterine contraction. So again, this is that positive feedback. It causes powerful rhythmic contractions of the uterine smooth muscle. And this leads to also dilatation of the uterus, which stimulates a greater release of oxytocin. And the oxytocin in turn again leads to contraction and dilatation of the uterus. So this is that positive feedback which leads to that explosive event, in this case, induction of labor or birth. And it has been actually speculated that this is the main hormone which is responsible for initiation of birth. Finally, oxytocin has a psychological effect as well. It's also called the love hormone, and it's involved in social bonding. It is involved in formation of a bond between sexual partners, or even between the mother and her newborn. So it also has these psychological effects. So that's it for neurohypophysis. And that's the end of my lecture about the physiology of endocrine system. So I hope you guys enjoyed it, and I hope you learned something new. It would be nice if you could fill out this feedback form so that we know what we can improve in the lectures. You can also leave a comment below this video or find me on social media if you have any questions about the topic I have covered. Thank you for your attention. Good luck with your exams and take care. Bye.